Okay, so this chapter is going to be on energy and nutrient relations. So the flow of nutrients as they go through an ecosystem, through organisms, through predators and prey, through herbivores and plants is all very important for how an ecosystem can function. Oops, sorry. All right, so of course, you have learned in basic biology that the organisms in this world can be organized according to trophic levels, and we can separate them according to these food webs as to where they fit on these trophic levels. And it isn't always, one step or another, but the most simple form or the base of all these food webs are autotrophs, which use inorganic sources of carbon and energy. So they get their energy from the sun, they use carbon dioxide from the air, and they form sugars. That is photosynthetic. There is also chemosynthetic autotrophs, and they use inorganic molecules as a source of carbon and energy found at hydrothermal vents, or really places of intense heat that have chemical reactions which they can use byproducts of to, again, make sugars. Heterotrophs are all other organisms which cannot photosynthesize or chemosynthetically produce sugars, and they then break down the molecules of other organisms that they have to consume in order to get energy. So that would be everything but plants or photosynthetic organisms in the food web. So the photosynthetic autotrophs can only use a specific part of the light spectrum. So I have the light spectrum down here shown before. You have right next to the visible light, so it's been kind of shown, broken out here, you have ultraviolet rays, which are much shorter and much more powerful. Now, the reason why plants or photosynthetic organisms don't use ultraviolet rays is because they are basically too powerful. There's too much energy in there, and it will damage the tissues and things trying to use it for energy. And infrared rays then are a little bit longer than that visible light spectrum, and they is usually felt as heat, and so it can increase the kinetic energy of air and other organisms. But they are weak. Infrared is weak. But there is a spectrum between the two which is just right. So kind of like three little bears. Ultraviolet, too powerful. Infrared, not powerful enough. The visible light spectrum between 400 and 800 nanometers. Or here we got 380 to 780. That's the spectrum of light in which it is used for photosynthetic activity. So this is called the photosynthetic active so not all of that light from sunlight, not all of sunlight is available for photosynthesis. But we can measure how much is available by measuring what's called the photosynthetic active radiation, and that's the number of photons per square meter per second, and this is called the photon flux density. Okay, chlorophyll absorbs light as photons. Photons are little packets of energy. But how these plants do that, or photosynthetic organisms do that, depends on the landscape around them. So much of it reflects, 10% of it, as you can see in this figure, reflects, 79% is absorbed in the canopy, in the vegetation above, 7% is absorbed in the middle of the canopy, 2% is absorbed by lower vegetation, and 2% actually reaches the forest floor. So 98% of it doesn't even actually reach the forest floor. Okay, but this 2%, this 7%, and this 79% is absorbed by plants and leaves and most of it is used for photosynthesis. Now, there are different ways that plants can photosynthesize in the way that they make the sugars. One of the ways is called C3 photosynthesis, done by C3 plants, and this is used by most plants and most algae. What happens is carbon dioxide mixes with ribulose biphosphate, bisphosphate, which is a 5-carbon sugar, and a phosphoglyceric acid, which is a 3-carbon acid, and that allows the fixation of carbon. And in order to do this, you have to have open stomata. When stomata are open, these are the pores in the leaves of the pores in leaves. This also provides an avenue for water to escape into the environment. So this process can cause water loss. So there needs to be a balance there. All right. So C3 photosynthesis, you get carbon dioxide, which mixes with rubulose bisphosphate to form PGA, which is a C3 acid, a 3-carbon acid. That goes through a different cycle to form sugars, and the end product is ribulose bisphosphate, which can then be used for another sugar molecule. All right, C4 photosynthesis then is another way in which plants can absorb carbon for photosynthesis. It uses a different enzyme called PEP carboxylase, which has a high affinity for carbon dioxide. So, because it has a high affinity for carbon dioxide, it does not need as many stomata to be open, so less carbon dioxide is needed, and also less water loss is there's less water loss. So when you combine PEP carboxylase with carbon dioxide, you get a 4-carbon sugar instead of a 3-carbon sugar, which is why it's called C4 photosynthesis. Acids produced during this fixation then are sent to specialized cells surrounding the bundle sheath, and then that breaks down this carbon dioxide, breaks down into a molecule of carbon dioxide and a 3-carbon molecule pyruvate, which then goes back to the mesophyll cell to form PEP carboxylase. That carbon dioxide then combines with ribulose bisphosphate to make the sugar just like before. Okay, and this is the process which I just explained. Okay, so you have PEP. The PEP carboxylase is the enzyme which adds a carbon dioxide to that. So now you have a 3-carbon sugar being made into a 4-carbon acid. That then moves to the... It then
breaks into a carbon dioxide and pyruvate, which then is converted into PEP carboxylase in the mesophyll. That carbon dioxide is used in the C3 acid cycle to make starches and sugar. So this is a way, by separating this is a way to reduce water loss by using less stomata and have a higher affinity for carbon dioxide. Another strategy is called CAM photosynthesis, or crucilation acid metabolism, and this is limited to succulent plants like cacti found in arid and semi-arid environments. Carbon fixation is the same as C4 plants, but it occurs only at night. So the stomata only open at night, so you lose oxygen there. And, you only lose water at night when it's cooler, so there's less loss. Okay, so this is a big cactus here. It, in the daytime, closes off its stomata, so it doesn't lose water, and then during the daytime, it uses carbon dioxide that it stores as this C4 acid at night. So at night,
it's open, carbon dioxide comes in, pyruvate combines with PEP, and carbon dioxide is stored as with this C4 acid. In the daytime, that C4 acid releases the carbon dioxide and sugars are made. All right, chemosynthetic autotrophs were discovered in 1977, living in the seafloor in these hydrothermal vents. Here's a map of where all these hydrothermal vents, which are between these tectonic plates. The bacteria then are these chemosynthetic autotrophs, and they use different chemicals and energy from them and the reactions taking place at these hydrothermal vents to fix chains of carbon, make sugars. This is then used by a variety of organisms, and these places are actually quite diverse for being in the ocean, especially at the bottom of the ocean. So here's a bunch of polychaetes which have aggregated there, and you see a lot of diversity of other species there as well. All right, heterotrophs. There's 3 feeding methods of heterotrophs. You got herbivores, feed on plants; carnivores feed on animal flesh for the most part; and detritivores. You also have omnivores, which are a combination of the two. Detritivores, which feed on non-living organic matter. Plants, animals, everything needs between 93 to 97% of these main elements: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. You look at the macromolecules, they make chains of carbon and hydrogen with occasional nitrogen, which is found in amino acids to make proteins. Oxygen is found in R groups in amino acids. You have phosphate groups, which are important for nucleic acids, and then sulfur scattered in between, which has some important parts in amino acids and other things. You also have other nutrients which are essential for life in plants: potassium, calcium, magnesium, sulfur, chlorine, iron, manganese, boron, zinc, copper, and molybdenum. And these are not needed as much as those other 5 elements, but they are still needed for normal functioning. So herbivores, they have lots of food available. However, the food that they eat is usually very low in nutrition, and usually does not have lots of protein content. So it's low in nitrogen, and nitrogen is needed for the making of proteins. So in addition to that, herbivores also have to overcome these physical barriers, such as plants have a lot of cellulose and lignin and silica, which are hard to digest and break down. So usually they have to form some sort of symbiotic relationship with bacteria or fungi to help break them down. And then a lot of them also have toxins in them, chemicals, which reduce the ability of them to be eaten, can sometimes be poisonous and kill animals as well. Detritivores consume dead and decaying matter and are very important for the breakdown of these dead and decaying matter and releasing nutrients into the environment. However, a lot of them, the dead and decaying things that they eat, aren't very nutritious, may have very low nitrogen content, and may still have a lot of these chemical defenses present. Carnivores, on the other hand, consume nutritionally rich prey, usually prey high in protein, which is usually very easy to break down and use for energy or make new proteins out of. But they can't choose prey at will because some other organisms have defenses, poisons, and other things which will prevent them from being eaten. Other organisms have found ways then to kind of mimic the looks of these poisonous organisms so that they also don't get eaten. So aposematic coloring is this bright coloring, which is kind of a warning signal that, hey, you can see me, you could eat me if you want to, but I'm poisonous, so you don't want to eat me. So these bright colors are indications of that. You see on bees, any sort of marking which makes them very distinct also kind of displays that they are not good to eat. And then you may have mimicry. So Mullerian mimicry is mimicry among several species of noxious organisms. So these poison dart frogs from South America. All of them have these really bright colors. All of them are really poisonous. And then you have Batesian mimicry, and this is where a non-noxious, harmless species mimics a noxious species. So you have this with coral snakes. Coral snakes are very poisonous, and they have red
banding across them. So that's a coral snake. However, you have these milk snakes, which are not poisonous, which have a similar banding pattern, although they don't have the yellow on them. But they are not poisonous. But things in areas where you find both of them, they will not be eaten because they look too much like the poisonous ones. Predator and prey. Well, predators, by selecting against these weak individuals, then set up, just by selection, the ability of these remaining population to have defenses against their predators, right? So there's selection against the weak, but those that remain usually have some sort of advantage in staying alive and can develop more. So this, in turn, selects against predators to have some way to get over those defenses. So what you end up is what they call a co-evolutionary arms race. And some examples of that are the cheetah and the gazelle. Both are very fast, right? Only the fastest gazelles get away from the cheetahs, or only the slowest gazelles get eaten by the cheetahs. So they're selecting against the slow and weak ones, and what's remaining are the fast ones. Well, in order to get more gazelles, then the cheetahs have to get faster. So the faster the cheetah you are, the more gazelles you eat, and so on and so forth. Similar examples are between garter snakes and these newts, which are very, very poisonous, but the garter snakes have developed a resistance to their poison. Bats and moths as well. We'll probably go over that in class. So the potential rate of energy intake by animals shows a relationship between the fooding rate and the food availability. And there's a limit on the potential rate of energy intake by plants, and that's been demonstrated by studying the response of photosynthetic rate to photon flux density, which we talked about before. So I have a little graph here that kind of shows as the amount of radiance level, so the amount of photons being emitted or absorbed in an area, it increases photosynthesis, but only to a certain rate. And of course, this can be different depending on different types of plants. It's usually very slow or at first, and then kind of levels off at a certain point at high intensities. Okay, and then this is a more general curve of the same thing. Pmax is the maximum rate of photosynthesis, so at a certain point, ISAT, that is where it doesn't matter how much more intense the photons get, you're still going to level out the amount of photosynthetic intensity. Holling described three basic functional responses in animals. One, in feeding rate. One is that the feeding rate increases linearly as food density increases. So food gets more and more and more and more abundant. Feeding rate increases more and more and more, only till a certain point. At this point, they just can't eat anymore, right? And this requires little or no search or handling time. So this would be like, there's just so much food and it's so easy to get. But this changes with search and handling time. So the second one is feeding rate rises in proportion to food density, but the feeding rate is limited by search and handling time. So at this point, it's not linear, but as you get more dense, you increase initially, but then over time it does also level off. And then the third one, which is that one down below, the feeding rate, there is an increase at low prey densities. It's very hard to find and handle prey, but as they get more and more dense, then there's a very steep or rapid increase in the feeding rate until it levels off.
Okay, and this is more S-shaped. So optimal foraging theory is that if there's a limit on the energy supplies, organisms cannot simultaneously maximize all their life functions. So if there's not an unlimited amount of energy, then they've got to allocate, or principle of allocation, between their demands for resources, whether that be survival, reproduction, or anywhere in between, or foraging, so on and so forth. So if everything else is equal, more abundant prey is going to give you a larger return. But you have to consider the amount of energy expended to get that prey, including search and handling time. So they will maximize the rate of energy intake based on the amount of time they have to search and or handle the prey. So an experiment was done that looked at bluegill, which are a type of fish. There are lots of small ones that are abundant. But optimal foraging theory would be, well, these larger ones are going to be, a better, easier to catch and more nutritious if you eat them. But if they get too big, then they're going to be too hard to catch. And so the prediction was that somewhere around 4 millimeters or larger would be where that optimal foraging theory would be. And when they looked at the real data, they found that that was the case. Same thing for plants, or a similar thing, which is that plants are going to spend, all other things being equal, the amount of energy needed, or the most, on the most limiting nutrient. Okay, so that may be calcium, phosphorus, or nitrogen, depending on what's in the soil.